All right, everyone, we're going to get started with our next few sessions. We're going to hear from our global research network, who has been dedicated to PURA syndrome research for many years. To start off, we're going to welcome back Dr. Hunt to the stage.
Great, thank you. So here we go. So I am going to talk to you a little bit about the PURA Syndrome Global Patient Registry. I've also been asked to talk a little bit about the Biobank. I'm not directly involved with that. The Biobank is based in Germany, but Professor Niesing's given me some slides which I've incorporated at the end. So most of the presentation will focus on the patient registry, and hopefully I will convince you all that you need to provide some data. So basically, the patient registry is part of a natural history study, and I'm going to explain to you why natural history studies are useful in understanding rare disease. Going to talk a little bit about the setup of the patient registry. And I do apologize to anyone who saw the talk from last year, because many of the slides are the same at the front end. I'll give you some numbers about recruitment to date and some of the issues that we're facing. And I'm very keen to hear from anyone in the room in terms of their experience of trying to enter data and whether there have been particular barriers. I'll talk about the integration with the Biobank and show you a few slides that Professor Niesing has asked me to share. And I'll finish on a slide showing you how to register if you've not yet done so and are inclined to. So natural history studies are important for various reasons. The main thing is that rare diseases are relatively poorly understood, especially after they're first described. And this sort of harks back to what I was saying earlier on, when in 2014 we started out with just the 15 patients, and we knew that that did not represent the full phenotypic spectrum of PURA syndrome. And basically, when we think about rare disease, the worry is that with small sample sizes, we're missing the less common, the rarer manifestations of a rare disease. And I think that the best way to think about this is that if you wanted to get a sense of mankind, we probably wouldn't accept a sample of a few dozen humans as being completely representative of mankind and all of human disease. So we'd need potentially tens of thousands of people in order to even get a sense of the most common diseases that affect us as a species. So when we're thinking about rare disease, we're obviously thinking that PURA syndrome has some core features, but it probably has much less common features that are enriched in the condition. We will only identify them if we have large numbers of patients and we follow them over a long period of time. I put as a second point there about ascertainment bias. So those very early papers that are published tend to be biased towards patients who've met criteria to be entered into research studies, for example. So often you miss the ends of the spectrum, particularly the milder ends of the spectrum. And of course, a lot of the genetic testing that we do takes place in children. So adults tend to be underrepresented. So we don't have a great deal of data about the long-term prognosis, which is obviously very difficult when you're meeting with a family to give them a diagnosis. And you have to say, well, actually, most of the data that we have comes from people under the age of 25 or 30, and we don't really have a good understanding of what happens beyond that. So I talked about rare manifestations. Obviously, long-term detailed data is what we really need in terms of deep phenotyping, so understanding what sort of problems can happen at what point in life. So through good quality natural history studies, it can be an effective way of identifying important quality of life issues and care needs. These are the sorts of things that can influence management guidelines, so we may well end up producing potentially evidence-based guidelines for particular assessments or surveillance. And at the moment, we, in our Gene Reviews article, have just made recommendations for what clinicians should think about. They're not strongly evidence-based. They're based largely on anecdotal data. So with a robust set of data, hopefully we will be able to produce much better statistics that will support recommendations. And one of the things that we talked about a few times during the question and answer session earlier was about genotype-phenotype correlations. Does having a specific type of genetic change mean that you're more likely or less likely to have certain clinical manifestations of the disorder? And also, where in terms of the position of that genetic change in the gene, does the position of the genetic change in the gene influence what we think in terms of what this may mean for an affected individual? So the patient registry was sort of conceived of way back at that first meeting. We realized that we needed to describe as many patients as we could and try to follow them up over a long period of time. But it isn't easy to set up a proper patient registry, and we were quite keen to do it as a digital undertaking rather than to try and collate things on paper. And so we were looking for partners to help us with that for some time, and then had to go through quite a prolonged ethics process in order to set up the registry. But this is the best way to systematically collect good quality data. And we're confident that if the registry is used, then we should in time be able to hopefully generate useful output that is beneficial to clinicians all over the world who are managing patients with PURA syndrome. So it has been made possible through a number of generous donations from private individuals and also from the PURA Syndrome Foundation. It is based in Southampton. The official name of the registry is quite long. It's the PURA Syndrome Longitudinal Natural History Study. So that's what's on all of the ethics documents, and that's what's on the consent form when you log into the registry. It's led by Professor Borelli, who is based at the University of Southampton. And we finally approved research ethics approval from the Health Research Authority back in 2019. Obviously, with COVID, there were some delays in terms of essentially launching the patient registry. But it is hosted through a partner at the University of Southampton. So that's the Clinical Informatics Research Unit. They specialize in hosting research databases. And as I mentioned earlier, the database was designed for healthcare professionals. So it isn't the sort of interface that most people would be used to in terms of logging on and navigating. All of the data that we collect is sort of managed through this company, Fonds Vision in Netherlands, and they provide the secure embedded database that underpins the patient registry and all of the projects that happen through CIRU. So the way that this works, it's quite straightforward. So it's a web-based application. So you go to the URL and enter a username and password that would be authorized once you've made an application to join the registry. It's really, really simple to do. You could do it in the time that it takes me to talk through this slide. It really is a matter of just emailing this address, pura@sutton.ac.uk, and all you need to include is the name and date of birth of your child or ward and country of residence. And this account is monitored by our research nurse, Tessie Thomas. I will forewarn everyone that she is not back in the office until Tuesday, so no one would receive a response before Tuesday. And then, provided that you provide those details, she will then grant you access to the registry with a username and password. And so the instructions for access, including a document for how to create profiles and go through the consent process, is included. And it is just a walkthrough process, but it is a few pages long. So you're not immediately enrolled in the study from the moment that you express interest. You do have to log into the site. You have to create a profile for your child. And once you've done that, then you will go to a consent page where you have to confirm that you've read the study documents and that you're happy to be part of the project. So first thing that you have to do after you've created a profile for your child is to then provide some demographic information. And only once you've done that does it open up the 8 existing modules, which are as listed there. So it's built as a system so that we can have more modules over time, and the plan was to do just that. There is a parallel arm for clinicians so that they can provide supplementary data for patients who are in the registry. And so when you go through the demographic module, you'll be asked to provide details for clinicians involved in your child's care who you are happy for us to contact. But we haven't been doing that actively at the moment. We've been trying to build up participation of families first. So in numbers, it's a little bit disappointing, if I'm honest. So the registry launched back in January of 2022, and we've been going for quite some time now. The green line is the trajectory that we were on. I urge everybody to join up last year, or at least to think about it, and you can see that the trajectory has sort of leveled off a bit since then. So in the past year, it's been very, very slow in terms of accrual. We've had 1 new enrollment per month for the past year. So as of June this year, we have 80 registered users with 81 different patient profiles and consent for 77 of the profiles. But then no demographic information was provided for 5 of those. So only 72 could potentially complete any of the modules, providing us with any clinical data about their child. And as you can see, this is the number of modules that have been completed. So once people get to this stage, it's generally quite good. More than half managed to complete all of the modules that are in there. And it does take some time, to be honest. These are quite lengthy surveys. They're mainly radio buttons, drop-down lists. There are a few free text boxes, but you do not have to do it all in one go. You can just stop, save, and come back at a time that's convenient. And I would suggest that's probably the better way to do it. Otherwise, you will be sitting at a computer for a good few hours. So it definitely doesn't all need to be done in one go. But there hasn't been much of an increment year on year. So the overall completion rate is just over 50% and has stayed pretty much static over the past year. At the moment, we've got participants from 17 different countries and 5 different continents. I haven't shown all of the countries there because some of them are really quite small, and I thought that it might indirectly be identifiable. So I've only shown those with 2 or more participants. Gender split is very similar to what it was last year, so a slight excess of females with males. I haven't shown the age distribution, but as I mentioned earlier, all but 2 patients are under the age of 30, and 50% of those enrolled are under the age of 10. So what are the challenges? Well, the obvious challenge is we can't seem to make this an enticing prospect for people at the moment. And I think it really is important to, as a community, think about whether this as a resource is helpful. We are very keen to see if there's something that we can do that will make this more accessible to families and easier to access. And I'll talk to you about some of the things that we're planning to do to see if they help. But overall, there are sort of big drops at different stages. So in total, we've had a respectable 192 requests to join the registry since it launched. Access was granted to 188, so 98% of people who asked for access were given access. But of those, more than a third never even logged in to the system. And I don't know what the reason is for that. I don't know how long the lag was between requesting access, being granted access, but I know that Tessie, our research nurse, has sent more than 50 follow-up emails that have not been responded to. So I'm very keen to know if there's anyone who requested access, was granted access, and then decided that they didn't want to go any further with it for any reason, and what you know, what the thoughts are about how we might be able to increase engagement. But of those who did log in, so 117 logged into the system, 37 did not generate a profile and did not come back to the system. So we lost a third of those who actually logged in at that point. And then of those who did create a profile, as I mentioned earlier, 80 registered users, 81 profiles, but there were 77 profiles that proceeded with enrollment where the consent forms were actually completed. So for those who got to the stage of creating profiles, actually providing data was relatively high, but we're losing a lot of people from the point of expressing interest through to getting access and logging in for the first time. So really very keen to hear what you have to say about that. And then in terms of complete data, we have complete data sets for 42 patients so far. So of the 188 original requests for registration, that's a relatively meager 22.3%. So quite a low conversion rate. So what can we do to try and make things a bit better? Because we really do want this to work for the PURA Syndrome community. Well, we think that part of the problem is perhaps not having the option to do this with some guidance. So I think it has now gone live on the PURA Syndrome Foundation website, but there will be an option for you to make an appointment with either Tessie or Lynn. So Tessie is our research nurse who's employed in Southampton. Lynn is a retired research nurse who also has a family member with PURA Syndrome. She is working via the PURA Syndrome Foundation as a volunteer. And so what they offer to do for you is to talk you through the process at an agreed time. So it's an appointment, it will be a video appointment. You can then start the registration process, make sure you get to the point that you can enter data into the performers. If you need any help, if you so choose, you can share your screen with either Tessie or Lynn if you're happy doing that. Otherwise, they can share a screen with you, which is not the one that you're using because they can't upload data, but basically a mock-up screen, a sort of beta environment where they can show you what you need to do. So we're hoping that by having the option of making an appointment and actually committing something to your diary, that maybe this will increase engagement and make it a slightly less daunting prospect. Because as I say, it's a little bit clunky. It really isn't the nice, slick interface that we're all used to. And I guess that's just the way it was developed, bearing in mind that it wasn't originally conceived as a sort of platform that would be used by patients themselves. So if we could, I think an even better option would be to not rely on families to upload the data if they chose not to, and to be able to do that for you in the context of some sort of structured interview. So basically talking with a healthcare professional, working through the performers, and with having them complete the performer on your behalf. But to do that, we do have to change our research ethics, which is not necessarily a quick thing to do, but it is something that we'd definitely be willing to consider if people felt that that would be a much better option. So the registry is tied up with the Biobank indirectly, so they're based on separate sites, but there are some interdependencies, and the Biobank is based at the Helmholtz in Munich. And there is a process map in place for how the two systems need to be navigated. And that is something for you to do. That's something for a clinician to do who wants to deposit samples into the biobank. So a corresponding profile does need to be created within the registry so that we have some corresponding data in terms of the clinical picture of the patient from whom those samples have come. So I showed this slide earlier. So as I say, these are two undertakings which do have some interdependencies. The biobank is something which is really important as a resource if we are keen to try to better understand PURA syndrome. We need a central repository of tissues and samples that could be valuable to researchers. The Helmholtz has an established biobank, so the PURA biobank will be essentially integrated into that. As I say, the patient registry that's in Southampton is built into a system that's used for research. So the two are sort of piggybacking existing infrastructure. And we have this process in place whereby there will be some linked pseudonymized identifiers, which basically give the clinical profile a match to the stored patient samples. So this is a slide that was kindly provided by Professor Niesing. So I showed this earlier as well. The plans for the biobank were developed back in 2019. The ethics was approved in 2022, and then in 2023, there were some serious problems at the Helmholtz with a cyber attack, and this completely delayed the launch of the biobank. And there were subsequently some other logistical things that needed to be considered, and the ethics, I think, had to be revised. But things are now on track for the biobank, I think, to go live relatively soon. Obviously, a launch date will be announced. The funding of the biobank is, in terms of running costs, is covered by the PURA Syndrome Foundation. They've also funded a part-time fellowship, and that's Simona Reba, who is helping to set up the PURA biobank there. And they have their own website, which you can check out as well. As I say, it is not live at the moment, the biobank, but there is information online if you want to learn a little bit more. And then just to come back to the registry, if I can, there we are. I do have to acknowledge quite a few people. So a lot of the work that went into setting up the registry was done out of goodwill through CIRU. The PURA Syndrome Foundation has also provided some financial support. We also had some generous donations to support setting up the patient registry and the appointment of Dr. Morby, who at the time was a clinical fellow, who basically single-handedly, or more or less single-handedly, Mel Anderson was also involved, went through this really arduous process of making sure that the ethics and data sort of protection standards were GDPR compliant. And so that took some time, but as I say, the system has been live now for quite a long time, and we just need to start using it, really. And there on the left is Tessie Thomas, our research nurse, who's the one who will respond to emails, and then Lynn, who is volunteering through the PURA Syndrome Foundation, is a retired research nurse, also willing to help with recruitment. So once again, if you do want to think about joining, this is the process. But I don't know if we have any time for questions at some point, but or just for comments, really. I'd be keen to hear what people have to say about their experiences if they've tried to join the registry.
Dr. Hunt, thank you so much for all your contributions. I want to apologize. I'm actually one of those who emailed, got the information registered, and never put in the information because honestly, it wasn't very user-friendly. And besides being a caregiver, it seemed very overwhelming. So the ease of just being able to get things in there, it just didn't happen.
Yeah.
So, but I'm excited now because also I was concerned about all of that data being in there so private.
Yeah.
So, but now that I have an understanding, now I'm eager to do that. So I want to apologize because all of this work you're doing for us, we need to do our part to help you guys. So thank you. And
I know I completely understand that life does get in the way. And this is one of the things that I thought was probably going to be the issue, is that, you probably, when you send that email, in the mindset of I'm going to do this, and actually, you don't get an immediate response with here are your login credentials. It could be several days, could be a week before Tessie gets back to you. I don't know what her typical turnaround time is on responses to join, but she does check the email at least twice a week, so she should be responding fairly regularly. But of course, I think when you've got lots of things going on and you know that this is something that's going to take a lot of time, and you look at it and you see pages and pages of things that you have to navigate through, obviously it's one of those things that's easily put off, and then life can just get in the way. And so I think that by having this option of diarizing something and saying, I'm actually going to speak to someone who's going to take me through this process and show me that actually it isn't as daunting as perhaps it may seem on the face of it, hopefully will translate into more complete data sets within the registry. So that's a hope. And we'll get a pretty good sense of that over the next 2 to 3 months, I think.
I was able to get like maybe 20% of the registry completed.
Okay.
Until I ran into it wanted like, what was the first genetic test done? And I was like, yeah, that was 14 years ago. I don't really know. So for us, it's a excess of data that I have to sort through her medical record to make sure that the information I'm entering in is accurate. Cause that's the point, right? So I think it will actually be really helpful to sit with Lynn in that appointment to help me navigate through this massive electronic medical record and say, where do I search for that information? How do I even access it from 2009? And which genetic test was it? And what did we have done? What metabolic test was done in the NICU? And all of those things that are like, I remember all of those things done, but I don't have the technical memory or expertise to like actually put that in as data for you, because there's so much.
No, I think you're right. And you absolutely can't do all of this stuff from memory. You do need medical records to hand, so clinic letters, discharge letters, things like that, that are going to help you to provide best quality data possible. And so it definitely doesn't need to be something that's done in one sitting, but I am hopeful that having someone talk through the process will just make it so much more accessible.
I was going to ask, is there any automated follow-up on some of these modules? Like our son is one year old, so a lot of those things he hasn't done yet and he won't do for some time. So we can enter everything perfect right now and it's only going to cover 1 year. Is there any
So
way to
I haven't
get back?
put that up here. It's remiss of me, but so the plan is to prompt people to update their data once per year. And then there'll be a sort of data freeze. So it's only by layering it that we actually get the natural history. So it's not a one-off thing. It is coming back to you each year and prompting you to update data so that we can build on what we already have and see how a person's condition is changing throughout their lifetime.
Actually, it's like a question is almost the same. My only concern is I know I did it, but I don't know what happened. I didn't get anything back. I know I did it the first time they send us something, and I know I did finish it, but after that I didn't heard anything anymore.
So you mean sending the email to Tessie, or you mean logging?
Yeah, I did when they first initially set it up and we got the email and stuff. I diligently put it all, whatever information was needed.
Yeah.
But then after that, I didn't get anything.
As a prompt to go back, you mean?
Yes, or
Okay.
if I did it right, or if I'm gonna get an
that-
update or something.
That is a good point. So there isn't a process at the moment to come back to you to say you've done this right. Thanks for doing it. So we probably do need to think about that so that people can be. No, at the moment, so you don't hear anything back, but that is a good take-home message for me, because we can actually think about that and communicating to say thank you for completing the registry. We'll be in contact with you next year. So yeah, okay, I've got it. Yeah.
Anyone else? Oh, Andrea.
Yeah, I was curious, are there other diseases that have registries that are you're able to just upload your electronic medical records to? Because I feel like that would take a lot of work away from parents, and then also like increase the accuracy of everything that you're
There are plenty of patient registries. So even within this registry, you're able to upload PDFs. If you've got lab reports, if you've got EEG reports, if you've got clinical letters, they can be uploaded. So there is a process for doing that. The problem is we don't have a way of extracting the data from a PDF. So it then creates a manual process at our end, but it is data that we want. But it is helpful to have it in a form where we have it embedded in data tables. So that would create a manual process for us at the moment. But yeah, I mean, it's important to gather that.
If I may add to that, there are companies out there that for some rare disorders, you can hire the company and they will collect the medical records and actually sort through some of it. And it's very helpful timing wise, but is incredibly expensive because this is incredibly time consuming, which leads to some of these issues. And if I may, while I have the microphone hijack for just a moment, I had wanted to jump in anyway and talk about the importance also of collecting your EEG footage and some other information. In discussion earlier, we were talking about the epilepsy and seizures in PURA can be a bit of an enigma. And I was talking with Dr. Nordley about how EEG technology has come a long way. And sometimes we can take old EEG footage and reanalyze it using new technology and learn stuff that we just didn't know before. Unfortunately, a lot of systems will cull their data intermittently. So an EEG done a year ago, your system may or may not still have that footage. Like we don't need the report from the EEG, we need the actual brainwave tracings. So if you have EEGs that were done recently, or you do some between now and the future, you may go through medical records and request the EEG on CD, because maybe someday we can learn more from that. And when designing clinical trials and so forth for, new medicines or for gene therapy and that sort of thing, you need to have some specific biomarker that you can go back and show, like, okay, you give gene therapy and the EEG looked like this before and EEGs after look like this. You need to have something to prove you can prove a difference, not just say people did better afterwards. So it'd be helpful to start collecting all that information now in case those trials come up in the future. So I would personally, when you have an EEG, I would ask for that footage on CD and keep that. And I would also start collecting your school records, like whatever testing was done through the school. I would start just compiling that and keep it so that someday when the time comes up, we're not scrambling to learn all of that information and gather all that information. We could try to have that ready ahead of time. Similar to the Biobank and the registry information.
Thank you, Dr. Hunter, for doing this. There is an opportunity, some of you probably know, NIH starting to do the similar study for many rare diseases almost 20 years ago, called the Rare Disease Clinical Research Network. They just announced the kind of RFA request for the application last month. It's due next month. So kind of continue this network, do exactly what every rare disease condition want to do. However, of course, so far about 20 consortium, 20 disease has got a benefit for last 20 years. So I think for the scale like a PURA, now you have a 500, 600. So it's became possibility to convince the research community. The key is, are you eligible for NIH as a PI for NIH or no?
I doubt it.
You doubt it, yeah. So it's a question that at the US, the community, like you're gonna have to convince some clinical investigator picking the PURA, gradually working on that one. So if you can join the consortium, that the resource will come. it's a money will come. It's very extensive money. And they usually give them one side per year, $1 million per year for 3 diseases. You have to choose in 3 diseases together. Then also a lot of other research to do this. I just say that just make sure that you all know this opportunity, particular foundation. So then you have to work in someone to starting to prepare maybe 3 or 4 years now. Now you have the strength and the capability to apply the grant, now get into some consortiums.
Questions?
Just one more, with regards to the polygenic nature potentially of the different phenotypes in PURA syndrome. I just wondered if having the raw data, raw sequencing data available to upload, like, is there a way to have that uploaded into the patient registry too? Or
Not at
how
the
do you
moment,
have
no,
access?
there isn't.
No,
No.
okay. Are there ways that other groups do that? I wonder.
There probably are, but not within this environment. Right?
Okay, gotcha. Okay. Excellent.
I was wondering because I know in a rare moment of productivity last summer, I think I filled out the first 4 modules and got caught up in my goal for this summer is to finish the rest. Is filling out partial of any help, or is it only kind of entered into the?
No, it is of help. I think the thing is that once all of those modules are completed, the plan will be not to complete them all again a year later, but essentially to review what was entered the year before and change things, and then it's basically frozen. So that's the way that it would be layered. So once you've gone through the pain of entering everything once, all that you need to do a year on is to review things and change things that are new. But having some of the data is better than not having any data. But ideally, having all of the modules completed is what we would go for. But we take some over none. Yeah.
Anyone else?
I'll just take a minute to point out, so in your swag bags, there's a nice little flyer in there about the registry, and there's a QR code that you can click. And what that QR code takes you to is the registry page on our website. And that gives you step-by-step exactly how to sign up for the registry. You'll also see a box at the top that gives you the opportunity to sign up for these one-on-one appointments with Lynn and Tessie. So that's available. And we also, Lynn actually went through and made a checklist for us. And you'll be able to find that on the website because it gives you an idea of what the information that you're looking for. And that way you can come to the registry with everything that you need before you, or I mean, you can go and enter it at little times here and there, but it gives you a list of what you need. And on that checklist, what we did was we talked to David and Diana, and they gave us key sections that if you're going to go in and fill out 4 sections this year, those are the first 4 you should do. And then the rest of them you can complete at a different time. So there's big stars right next to it that tells you which one. So please go to our website. It tells you step by step how to do it. And if you do make an appointment with Lynn or Tessie, please make sure you set your account up before you make an appointment with them so that you're just ready to hit the ground running. So thank you. All right. Thank you, Dr. Hunt. All right, next we're going to hear from another member of our Global Research Network, Dr. Robert Janowski. He is a staff scientist in the Institute of Structural Biology at the Helmholtz Center in Munich. He is interested in the structural studies of human PURA, its mutants, and their interactions with RNA. Welcome.
Thank you a lot. I guess it works. So first of all, I would like to thank Liz and Eva for giving me opportunity to come here and join you. It's my first time participating in a PURA conference. First time it was in Philadelphia. a couple of years ago, last year in Kingston, and now in Chicago. And a second thing I want to say, I want to apologize in advance if some of my slides experiments will be not very clear to all of you, but I promise I will be here till tomorrow, and if you have any questions, I will be happy to answer. So let me give a little bit more introduction about myself. So I'm originally from Poland, but after completing my PhD, I moved to Hamburg, to Germany, to the north. Afterwards, after a couple of years, I moved to do research in Spain, in Barcelona. And 11 years ago, I moved to Munich, to Helmholtz Center. So Munich is quite a nice city located very close to Alp mountains, 1 hour drive. I guess most of you know Munich as a city of fun, so-called Oktoberfest. It's a beer festival which happens in September, although it's called Oktoberfest. But Munich is also a city of many universities, many public research institutes, and a number of smaller or bigger pharma companies, biotech companies. And Helmholtz Munich is a public research institute, which is located a little bit far from the city center. So this is our campus. This is where I work. And today I would like to present you some of the results which we just published in eLife journal 2 months ago. So before I will go to the details of our research, I would like you to show this web page. It's called ClinVar, from clinical variant. And if you type the name of the gene, in this case, PURA, you will find the list of all registered or updated variants of PURA. And here on the right, you have a classification. So you can see here that some of them are pathogenic, some of them are benign. And our interest is to understand, first of all, correlation between the gene and so genotype and phenotype, and also to understand why one is pathogenic and the other one doesn't give so many bad symptoms. So that's why we want to understand it on a cellular, molecular, and atomic level. So in our group, most of people work on the PURA project, and we combine different approaches. So we try to test the localization of PURA in the cells, different type of cells. We check interatomic, PURA other proteins, PURA nucleic acids. We also do, where I'm mostly involved, structural analysis and biophysical analysis. And this is what I will be talking about. So just to remind you, so you already know that PURA is composed of 3 repeats, so-called PUR repeats, and this is the schematic view of the protein. So first 2 repeats form so-called nucleic binding domain, and third repeat is responsible for dimerization. So PURA protein is dimeric protein, and this is how it works. It must be dimeric to play the correct function. So this is a scheme of the dimeric protein. So as you see, this third repeat interacts with a second copy of identical repeat, and here in the middle of the protein forms interaction and dimer. So if we would like to look at the model in 3 dimensions, so actually this is what I do. I use experimental methods to see how proteins look in 3 dimensions, and also how they change if there are any changes on the amino acid level. So here you see one copy of the molecule in the gray color, and the second one in the same colors like on the scheme above. So once again, we have this nucleic acid binding domain of both sides and dimerization domain in the middle. So now I would like to show you the methods which let us understand, first of all, this is what we want. First of all, we want to understand how PURA protein works, what it causes. So first thing method which we use is, first of all, for this paper, for the results which I will show you, we selected some of the variants. Of course, we cannot use all possible variants. It would be really a lot of work. That's why we selected some of the variants located in all 3 repeats. And we know that some of them give really strong symptoms, while some other, for example, this one looks like it doesn't give any too much harm to the protein. So in the next slide, I'm showing first experiment. So we try to check how PURA behaves in vivo, directly in the cells. So for this purpose, we use microscopes, fluorescent microscopes, and we just check how the cells look like. And we have labels for PURA, so we can, with colors, we can identify where it is located in the cell, exactly in which compartment. So in this experiment, we tested how P-bodies. So P-bodies, it's the same as processing bodies. So we are such little bubbles which contain RNA and other proteins. So this is kind of protecting state. And in every moment when cell needs, this RNA from the proteins can be released and play whatever function it should play. So in these images here, all the little dots which you can look here, these are the processing bodies. And for this purpose, we compared wild type, so untouched puree protein, and compared with a few different variants. And we found out that so we can actually measure the size of the processing bodies and compare in all the cases, and then quantify it. So what we found out that the formation or association of the processing bodies is impaired by some of the mutations. So for the wild type PURA, we have, let's say, normal level of the processing body formation, while for the other variants, patient-derived variants is much lower. So definitely it will affect the function of the cell, because there will be a misregulation of, there will be misregulation of the RNA, which is stored in the P-bodies. So this is another method. So this is actually something what I personally can do. So here you have 3 different X-ray structure. We use X-ray crystallography to see how the proteins look in 3 dimensions. So this experiment, so it's not model, it's experimental model. So here I compare the RNA binding domain, just repeat 1 and 2 of PURA. This is the wild type. So this is how it looks in the healthy cells. And I compared with the variant, which is benign. So this is R140P variant. And when we compare both, there's actually no difference. So just from this, we can already guess that this variant should not harm the cells, and it should not give any strong symptoms to the kids. While in contrast, for the variant K97E, we see that the protein structure, this fragment, is impaired. It's totally messed up. So although the mutation is located here, shown in red, somehow it affects the other part of the protein. So here we have shift of the amino acids. So although the fold, like the shape of the protein, is still, it still looks the same, the electrostatic potential of the protein is changed. So this is what I'm gonna explain on this. So we can also, there is different way to show the protein structure. We can show it as electrostatic potential. And as you know, plus always like to interact with minus, opposites attract. So we can show the protein structure in blue color where there is positive charge, in red color where there is negative charge. So this is positive charge, and nucleic acids are negatively charged. So it means, and we know exactly that this is the area where nucleic acids, DNA and RNA will interact. So you see the first two, they are kind of identical, but where we look at K97E mutant, you see that there are much more red areas negatively charged. So it means that binding to RNA or DNA will be changed. It will not be the same as for the native protein. And this is what I'm proving on the next slide. So this is, I'm sorry if it's too difficult, but I will be explaining later if you wish more. So this experiment is called EMSA, the electrophoresis mobility shift assay. So I will explain on this one how it works. So we have polyacrylamide gel, and we load on the top here RNA with increasing concentration of PURA, native or whatever variant we would like. And we run through this gel electrical power. So the samples will migrate depending on the size. So if there is no interaction with RNA, we will have RNA sample on the bottom here. But if PURA interact with RNA, it will be heavier, so it will migrate slower, so it will stack somewhere here. And here you see for the 3 different variants. This is Y-type, R140P and K97E. So maybe from this, it's not very obvious how much protein interacts with RNA, but when we quantify it, when we measure exactly intensity of all these things, we can easily quantify it. So this is quantification of all 3. So if we assume that the native untouched healthy PURA interacts with 100% with RNA, you see that the variant K97E has just 50% of ability. So it can just catch 50% of RNA molecules. Okay, let's go to another experiment. So you know already that PURA can interact with RNA, but I guess not everybody here knows that PURA has additional function. It can dissociate double-stranded DNA or even double-stranded RNA. So DNA likes to be double-stranded, as here on this scheme. So PURA has this power to intercalate here into the grooves of the DNA and separate to make it single-stranded. So 2 chains will be totally in different positions. So we can actually experimentally test if indeed PURA and the variants play the same role. So we label double-stranded, each of the double strands with some chromophores. So this one, when we illuminate it, it starts to emit light, which we could detect. But if the second one is close to this, the second one will catch the light. So imagine we illuminate double strand and we will not get any light. But when they are separated and we illuminate it with special wavelength, this one will emit light, but this one will be too far to catch it. So we will observe emitted light. And we can mix such double-stranded DNA with different variants of PURA and see what's the difference. So here I show, first of all, the graph, how it would look when we measure fluorescence, the emitted light, with increasing concentration of PURA. So in blue, I show the Y-type, properly working PURA. So everything is fine. The variant, this benign variant R140P shows more or less the same behavior. But in contrast, variant K97E shows almost no unwinding. So because of all these changes in the structure, which I presented before, it has no power to interact properly with DNA and play the role it was designed for. So similarly, like before, we can quantify this. And as you see, if we assume that native PURA has 100 power to unwind DNA, then this mutant has only 10%. So you can imagine if this process doesn't occur in the cell, it harms the cell, and then we get all the symptoms at the end. Okay, and another thing which we do in our lab, this is this is experimental thing, but not in the laboratory. This is what we do using molecular dynamic calculations. This is all done by the computer. So because we know the structure of PURA, we can put the structure to the program and change one of the amino acids and tell the program, tell me what will happen if this amino acid is changed by something else, or tell me what will happen if one amino acid is deleted or shifted. So this is what simulate molecular dynamic method tells us. So at the end of the calculation, we always compare the untouched healthy PURA with the one which has some change, genetic variation. And then from the graph which we have, we can kind of guess what is the behavior of the whatever variant we would like to say, to check. So this is something what we could actually do for many variants, in contrast to the other methods, which are really time consuming and expensive. Okay, I was talking about RNA binding domain or nucleic acid binding domain, but now I would like to talk a little bit about the dimerization domain, repeat 3. As I said, we selected some variants, and for this investigation, we selected F233 deletion variant and also R245P. So first of all, here on the left, you see the structure, experimental model of Y-type PURA without any defects. And it looks like this. There are 2 copies of molecule, of identical molecule, with the alpha helices like swapped, giving it lots of stability. But what happens if we delete one amino acid? So this is position of amino acid phenylalanine 233. So if we delete one, so the next one has to occupy this position. The very next one shifts. So all the other amino acids shift by 1. So obviously it will affect a lot electrostatic potential. And as I mentioned before, electrostatic potential is something very important for the interaction. So first of all, this electrostatic potential will change the interaction here within the dimer. So probably this dimer will not exist. And it may also affect the interaction with other proteins and nucleic acids. And similarly, in the case of this variant, R245P, so this here we replace arginine by proline. So the arginine is located in a very extended part of the protein chain. So it has to be extended. And proline residue has this weird property that it's very rigid. So when we put it instead of arginine, this amino acid strand will not be extended, it will bend. So if it will bend, the whole structure may suffer, so it will not look like this. So probably dimerization will be also abolished. And this is what we confirmed with experimental, with the experiment. So for this, to check if PURA dimerizes, we used so-called nanobread method. So I will explain you how this method looks like. So we have one monomer of PURA, and we have second monomer of PURA, and we want to see if they interact. So one monomer is labeled with some extra protein, and this monomer is also labeled, it's attached to the protein, with another one. So when both interact, when they form dimer, the labels are also close to each other, and they can perform a reaction which will release some fluorescent signal, which we can measure. We can detect it with the microscope or detectors. And in contrast, if there is no interaction here, if the interaction is abolished, both labels will be far away, so they will not perform the reaction. So the signal, this fluorescent signal, will not be released. So we will see nothing. So this is what we actually observe for, for the variant. So here we have this emitted fluorescent signal. So this is our positive control. So this is like untouched wild type PURA, which contains untouched repeat free. So if we mix two perfectly fine PURA molecules, they will make dimer and we will record the signal. In contrast, as negative control, we used just repeat 1 and 2 of PURA copies. So it doesn't have dimerization domain. So it cannot dimerize. So it will not give any signal. And what we observe when we use any of the variants, when we try to dimerize it with the wild type PURA, you see that dimerization doesn't occur. Or if there is any, it's really weak and not for all the molecules. So dimerization can be also abolished by some of the variants which occur in the repeat free. And dimerization is also very important for the function of PURA. So I think I'm at the end of my talk. So the conclusion from the experiments which we've done and published are here. So as you see, the sequence variants which we tested can affect quite a lot of things. So first of all, they affect association of processing bodies, these bubbles of RNA, which are important for proper functioning. Defects in PURA can affect nucleic binding. They also reduce unwinding activity, the separation of double-stranded DNA. Also, if the variants are located in the repeat free, it may affect dimerization of PURA. And also variants, especially in the repeat 1, 2, and 3, can affect integrity of the structure. So the structure can unfold and do not play anymore the proper role. So this is my last slide. I would like to acknowledge all members of our group who are involved in the project, all the collaborators who also work with us, and once again, PURA Foundation for giving me a chance to come here. And I'm ready, and I would like to thank you for your attention, and I'm ready to take your questions. And I'm really sorry if it was too complicated to understand.
Okay. Taylor?
I have a hard time following all those numbers. Is the lower the variant, the better it comes out to be?
Lower variant.
As in like, if the number is 97 with a P compared to 253, is it going to show any difference?
Well, difference, yes, but it doesn't mean, you mean the number in the sequence, the position in the sequence?
Yes.
No,
Okay.
no, because we have, as also David mentioned, the cases where, okay, maybe I will go to the slide. Can I go back? Yeah. So actually, it doesn't matter. This is the problem with PURA protein. It's really behaving in an unusual way, because in any other proteins which also are affected by some genetic variations, usually all the mutations, the problem defects, they accumulate rather in one specific region of the structure, while for PURA they are spread everywhere. They are really spread, so they can start from the very first amino acid, and they can occur here at the very end, and all of them can give really severe symptoms. I would rather say that defects in the well-defined parts of the domains are, I would say, they are more into giving severe symptoms than the ones which are, for example, in some areas of the structure which are less important for the function. But I would say to really give statement like this, I need like much. I hopefully we will not get much more cases, but only with higher number of cases, such hypothesis or conclusion could be made. But generally, it doesn't matter where that occurs. It can give severe symptoms. Yeah.
Hi, when you're showing the EMSA data, the
EMSA.
yeah, can you bless that, please?
Slow. Yeah.
In the R140P, it looks like at higher concentration, there's actually more RNA binding, but it doesn't look like that when you quantified it.
Yeah. Actually, this is just the exposition of the gel, so it has nothing to do with the amount which was shifted. Because at the end of the day, we measure all the intensity here and all the here. So it's not that we can compare one with the other one. So it's just coincidence, and here we have a little bit stronger signal, but in total, the amount which is shifted here is identical like here, or very, very similar. While here you see it's much less, so it's much more here, as you see, it's I think to show. So RNA here like mostly goes through the gel and doesn't bind. Yeah, even with the highest concentration, you see still RNA which is unbound. While here, with the highest concentration, all the RNA is already bound by PURA.
Okay.
Does it answer your question?
Yeah, thank you. I also had another question. Do we know if there's any dominant negative inhibition of the mutant PURA?
Could you repeat, please?
Do we know if there's any dominant negative inhibition of the mutant PURA protein?
What would you mean down?
Oh, dominant negative inhibition?
This you mean PURA inhibiting itself or upregulating this?
Yeah, like the mutant PURA protein. Do you know if it's inhibiting the?
I don't have any knowledge about this.
Okay. Thank you.
I have a question about so you tested the mutants in the nucleic acid binding for their ability to bind RNA and then their ability to help with the unwinding of the DNA.
Right.
And then in the dimerization subunit of the gene or protein, you tested the dimerization. Did you test the opposite? Like, did you do any experiments to look at the nucleic acid binding and the dimerization mutant and vice versa?
Yes, absolutely. So I didn't mention here not to extend my presentation too much, but maybe I will show the scheme. So dimerization domain also interacts with nucleic acid, more or less with the same strength like the other domain, and it also unwinds double-stranded DNA. So if we look at the model, so actually, we assume that the middle domain starts unwinding because DNA, double-stranded DNA, is kind of long. So we just assume that the middle part, this dimerization domain, actually starts unwinding, and then both parts help. And when it's already separated, it can actually bind on both sides of full-length protein.
Thank you for presenting all that. I'm also curious, what's next for your lab, for Dr. Niesing's lab? I know from past conferences that we've, implemented some of the CRISPR-Cas9, gonna try to say it, poly or stem cell, whatever, induced, blah, blah, blah. So I know that Dr. Nisa was working on that. I'm just curious if you have any like quick updates
So
or what's next? What's the next step?
So I'm not, I'm just involved in more or less structural biology part. So I'm testing the interaction with nucleic acid on the atomic level. So just try to get the three-dimensional model and to see where they exactly interact, which amino acids are important to conclude, again, how the variants will affect this binding, to see it on the atomic level. And with the cells, we have quite a lot of results, which I cannot present because it's not my work. But hopefully the next year we can give you some update with all the cells and other methods, proteomic, interactomic.
Yeah, I think we have one online question too.
We have one online comment. Excellent work.
Oh, thank you.
Okay, so where is it? What is the difference between all the PURA deletions? Can you answer that?
Well, it's
It was a
the
rundown.
question is kind of not really precise because it can be one deletion, it can be one amino acid deletion, like for 233 variant. It can be deletion of several amino acids, and it can be deletion of the whole copy of the gene. Our website, would that be? Yeah, I mean, this information is on the webpage I shown. But the difference, I mean, different symptoms probably, but if you must remember that there are two copies of PURA, and one is always healthy. So there is always 50% of the protein which works fine, and the other 50% is totally, completely missing if the whole gene is deleted, or some fragments of the proteins can be missing if there is a codon somewhere in the middle. But half of the protein has proper function. So this is what we should remember.
Do we know what kind of macro effects might influence how much of the protein is produced in the mutated allele? Could there be certain diseases, for example, that might affect whether or not there's any action at all on that allele?
So you mean, if I understand, you ask if there is the same amount of the PURA protein, the healthy copy and the affected?
No, I'm saying in the affected protein,
Yep.
there's something is being produced, right?
Yes, yeah.
Right?
Mm-hmm.
So could something in the state of the general body, like an infection, for example, influence how much of that protein gets produced, if any? Could it lower the functioning of the affected allele?
So if there is any control of the expression, it would affect both.
Yes.
It would affect both, not just one of them.
Okay.
Right. Okay, all right. Thank you, Dr. Janowski. Very happy to have you here.